Hello and welcome to our video on the cardiac muscle tissue. In this presentation, we will learn about the location of cardiac muscle tissue, the anatomical characteristics of cardiac muscle tissue, the functional characteristics of cardiac muscle tissue, and how it differs from skeletal and smooth muscle. Cardiac muscle tissue is found only in the walls of the heart, so it differs from the skeletal muscles that we are familiar with. Additionally, unlike skeletal muscle tissue, cardiac muscle tissue is more sensitive to low levels of oxygen. When skeletal muscle tissue contracts under low levels of oxygen, it can use anaerobic respiration and produce lactic acid as a byproduct. However, cardiac muscle tissue does not do this and is therefore more sensitive to tissue damage if it does not receive a constant supply of oxygen. Lastly, cardiac muscle tissue is made up of cardiac muscle cells which have excitable membranes, like skeletal muscle tissue. However, as we will see in the upcoming slides, cardiac muscle tissue differs anatomically and functionally from both skeletal and smooth muscle tissue. Cardiac muscle tissue is made up of cardiac muscle cells. Unlike skeletal muscle cells, cardiac muscle cells are small in size, are typically branch-like, usually have a single nucleus, but sometimes have a few nuclei, have short, wide T-tubules without triads, contain a sarcoplasmic reticulum with no terminal cisternae, are nearly totally dependent on aerobic metabolism by containing lots of myoglobin with many mitochondria, and contact each other through intercalated discs. The T-tubules are important for relaying the action potentials used for inducing a muscle contraction by exciting the membrane. The sarcoplasmic reticulum is an organelle used for storing calcium ions. Calcium ions play an important role in the contraction of the muscle proteins. As mentioned before, cardiac muscle tissue is highly dependent on aerobic respiration for metabolism. This is accomplished through the high myoglobin content that has a high affinity for oxygen, and through the fact that there are many mitochondria. Mitochondria are organelles that act as the site of aerobic respiration and provide a high amount of ATP, a high-energy molecule used for muscle contraction. Lastly, cardiac muscle cells communicate with each other through intercalated discs, which contain gap junctions that allow ions to move easier from one cardiac muscle cell to another. Intercalated discs are found only in cardiac muscle cells and are specialized connections that join the sarcolemus of adjacent cardiac muscle cells by gap junctions and desmosomes. Their functions include the following: stabilizing the positions of adjacent cells, maintaining the three-dimensional structure of cardiac muscle tissue, and allowing calcium and sodium ions to move from one cell to another so that cardiac muscle cells beat simultaneously in rhythm. Allowing the cardiac muscle cells to beat in rhythm allows the chambers of the heart to beat in unison, so that blood can be directed with a strong pumping motion, pushing blood from one chamber of the heart to the next, or from the heart to the lungs, or from the heart to the aorta, which then flows to the rest of the body. The unique characteristic of cardiac muscle tissue is its ability to contract without neural stimulation. This means that the brain does not need to relay messages to the heart in order for the heart to continuously beat. The contraction of the cardiac muscle tissue is initiated instead by specialized cells known as pacemaker cells. These cells are embedded in the walls of the heart in the right atrium. Some of these pacemaker cells are bunched together to make the sinoatrial node, SA node, which is the primary source for starting an action potential that causes the cardiac muscle cells to contract and the heart to beat. There is also another bunch of pacemaker cells, located inferior to the SA node, called the atrioventricular node, AV node. The AV node cells start an action potential after the SA node has already sent its electrical signal to contract. If for some reason the SA node fails to cause the heart to beat, the AV node can take over. But if the AV node becomes the primary pacemaker source, then the heart rate will be slower than if the SA node was still the leader. The nervous system innervates the SA node and can modify the heart rate and the force of the contractions by altering the pace and tension of the contractions when the parasympathetic or sympathetic nervous systems are stimulated. Contractions of the cardiac muscle tissue are 10 times longer than skeletal muscle to allow for prolonged expulsion of the blood from the heart, and there is a longer refractory period. The longer refractory period can allow a premature contraction to occur, which could result in irregularities of the heart rate, such as cardiac arrhythmias. The long refractory period is accomplished through the special properties of the sarcolemma of the cardiac muscle tissue, which prevents wave summation and tetantic contractions, which are things that can happen in skeletal muscle when we want to lift something heavy and hold it up for a while. But with the heart, those would not be beneficial features, because it needs to have a consistent rest period between contractions so that the 4 chambers of the heart can fill with blood. In summary, cardiac muscle tissue is located only in the heart, is made up of small, branch cells known as cardiac muscle cells, and is sensitive to low oxygenated conditions due to high dependence on aerobic respiration. Cardiac muscle cells contain intercalated discs that support cardiac muscle tissue both structurally and functionally. Cardiac muscle contraction is involuntary, like smooth muscle contraction, and not voluntary like skeletal muscle contraction. In order to function, cardiac muscle tissue does not require stimulation from the nervous system and instead can use pacemaker cells to control heart rate. These specialized pacemaker cells control contractions of the heart to prevent heart rate irregularities. Here is a quick reference chart of some key features to help you remember the differences between the 3 different types of muscle tissue found in the human body. These 3 types of muscle are also found in many other species of animals as well. Thank you for watching, and have a great day.